Hello everyone, today I am going to be discussing the science of bread and the four basic components to a bread that rises as opposed to a flatbread. So a bread that rises typically includes flour, water, yeast, and salt as the very essential ingredients. If you have a flatbread, for example, you can make a very basic flatbread with flour and water, or flour and water and salt, but it's not going to have that rise that yeast provides. And also, if you exclude salt, then you're not really going to have a very good flavor. And if you don't include water, then that's just, it's not even going to come together. So all of these four ingredients are very, very essential in order to make a basic yeasted bread. And today I'm going to be discussing essentially the bread making process a little bit and how these four main ingredients play a role in how bread is made. Getting into nitty-gritty of some of these things, but not nitty-gritty of everything. And I'm mainly going to be talking about bread that's made with store-bought or commercial yeast as opposed to sourdough. So who am I? Why am I talking about bread? I've been pretty obsessed with bread for about 4 years, getting into sourdough, getting into different types of breads, whether they're sweetbreads or croissants and different derivations of breads, pretty much going as crazy as you possibly can with bread. And I have a very long ways to go. So I wouldn't qualify myself as a professional in any sense of the way. I'm a home baker, and I'm also a soil scientist in my professional life. So obviously the science of bread becomes a bit more intriguing if you think about science on a daily basis. So at least for now, I'm planning for this video series to be 3 parts. There might be more in the future, but I'm not sure. Today, of course, I'll be discussing part 1, and then in the future, I'll also be getting into the bread making process, probably starting with a normal yeasted dough, and then possibly an introduction into sourdough bread, and then I might go further and deeper into that. So in our discussion of flour, water, yeast, and salt, first I'm going to discuss flour, and I'll focus today specifically on wheat flour because that's pretty common around the world for making bread. And that discussion will be broken up into 5 parts. The first part being the grain itself, and then the milling of flour, and then the starch that's in the flour, the protein, and the enzymes, because those are very important components of flour that contribute to the bread making process. So the seed usually contains everything that the little baby plant needs to start germinating and starting to grow. And then after that, it starts to utilize some sources from the soil and water and the atmosphere in order to grow. We can think about it in 3 different components. We have the bran, the endosperm, and the germ. The bran is, of course, that fiber-rich outer layer that protects the seed, and it also contains really important B vitamins and trace minerals. The endosperm is the really starchy component of the seed, and it also contains the protein, which forms gluten, and we'll be discussing that later. And then we also have the germ, which is that little orange section, and this is just a little nutrient-rich core that contains antioxidants like vitamin E and then more B vitamins, as I discussed in the bran, as well as healthy fats. So another important thing that the seed includes is it includes enzymes, which can help break down the starches, proteins, and fats within the seed as it grows into simpler substances that the plant can use for metabolism. So if we continue our discussion of the grain itself, about 100 grams of whole wheat flour, which is flour that's ground from the whole wheat grain, usually has about 340 calories, about 13 grams of protein, about 72 grams of carbohydrates, which mainly are starch within that carbohydrate fraction. And that's considered sort of a complex form of sugar, and we'll be getting more into that later. We also have less than a gram of sugar, about 11 grams of fiber, and about 2.5 grams of fat. So if we look at the pie chart on the right-hand side, most of what makes up the flour in this case are carbohydrates, followed by protein. And those two sectors of the flour are quite important in the bread making process. So now if we get into talking about the process of milling, which is when those grains of wheat are milled into flour, there's quite a big distinction between whole grain flour and all-purpose or bread flour. And what that is that the whole grain flour is typically made from the whole ground up grain of wheat, meaning that it contains the bran and the germ, and it contains the nutrients and the fats that are located within those 2 components. So essentially whole grain flour is a bit nutritious and also more fiber rich. as compared to all-purpose or bread flour, which is usually made specifically from the endosperm, which is the part of the flour that's most enriched with carbohydrates and with proteins, but also lacks those vitamins and minerals and that we can get from a whole grain flour. A big difference between the two of these in bread making is that even though the whole grain flour is more nutritious, the fact that it's whole grain and has those bits of bran in it can really affect the texture of the bread and the gluten development, because, for example, if you have these bits of bran in the flour, it's going to weigh down the dough and also interfere with the structure of the gluten, and it can essentially create a more dense bread as opposed to an all-purpose flour or a bread flour, which tends to make a really light and fluffy bread. Now let's get into starch, which is a pretty big component of flour. Starch is also a really big component in other things that we like to eat, such as potatoes and rice, things that are usually very delicious and things that are usually the base of most foods throughout the world. Starch is very important in flour for a few things, as well as helping to absorb water, and it goes through a process of gelatinization during the baking process, which helps sort of solidify and form the bread structure, which also helps it trap the CO2 bubbles that form during fermentation. And in addition to that, another really important thing that starch provides in bread is flavor. When I mentioned earlier that if you just cook a flatbread that's made from flour and water, it's not going to taste very good. But with bread, it usually has to rest for a while and go through a process called fermentation, where some of the starches start to break down, and that releases sugar. And sugar, of course, is delicious, and then the flavor becomes more complex. So essentially, through the process of bread making, you're extracting a lot of flavor from the starch. So along with starch, gluten is incredibly important in the bread making process. In terms of protein percentages, in the US, protein makes up about 10 to 15% of most wheat flours, and in Canada it makes up a slightly higher percentage, about 13 to 16% of flour is protein in Canada. And the important thing about the protein in the flour is that it is in two components. We have gliadin and gluten. So you think of flour, and technically flour does not contain gluten. It contains the building blocks for gluten. And the way that those two building blocks combine is usually via hydration, which means just to add water. And that water helps these two components of gluten join together with these hydrogen and disulfide bonds and helps them stick together in a line, and then that helps to sort of build strength and structure within the bread. So if we think about the gluten development process, as I mentioned before, hydration is very important. If you see this figure on the furthest left-hand side, the first step of gluten formation is hydration. But then what's happening there is that the gluten strands are forming, but they're quite bunched up. As you can see, they're quite wavy. But then through a process of stretching, also known as kneading, and also relaxing the dough, those gluten structures start to stretch out, they start to align better, and you start to get more strong sort of gluten chains that help really form the structure of the bread. If you see the middle GIF here with the person sort of gently stretching and pulling the dough, that is a technique called the stretch and fold, which is a way to very gently stretch and develop the gluten. And this is usually done in sourdough breads, which take a long, long time to ferment. And usually during the fermentation, the gluten is gently developed over time, as opposed to the figure on the furthest right, where the person is rigorously developing the gluten through some very vigorous kneading. So that's sort of a fast way to develop gluten, and it's very effective, of course. So another important thing about gluten and developing it is that after the kneading process, if you let it just rest, you'll notice that after 30 minutes or longer, if you try to stretch it again, it'll be much more stretchy and elastic than it was right after you finished kneading it. And that's usually because with time, the gluten can better hydrate, the flour can hydrate a little bit better, and it also helps it relax. And so that's where the stretchiness comes from. So this is the first slide of fast gluten development, and then the second slide I'll be talking about slow gluten development. So as mentioned, for like the fourth time in a row or something like that, hydration is key in gluten development. So if we think of a fast dough, which is what I would call like a commercial yeast dough or a regular yeasted dough made from store-bought yeast, Kneading is very important. You want to, after you've finished combining the ingredients, which you can see in the left-hand image here, even though the dough is quite shaggy and doesn't have a lot of structure and doesn't really hold together well, after the kneading process, which usually takes about 10 to 15 minutes, we have the dough on the right, which is the same dough, but it's really nice and smooth because we've really stretched it and developed those gluten networks, helping to create this nice tight ball of dough with some degree of elasticity. So the dough that I discussed in the previous slide did require a short fermentation time of only about 2 hours, and therefore it did need to be kneaded in order to develop that gluten structure in such a short amount of time. But in this slide here, we have what is called slow gluten development. And in this case, we can have a dough ferment not for just 2 hours, but even 12 to up to 24 hours. And in the picture on the very left-hand side, all I did was just mix the dough together. There was no kneading involved. And then I just covered it and let it rest for 22 hours. And as you can see, after 22 hours, it's very bubbly and puffy and active. And then at that same time, as I was pulling the dough out, you can see these sort of stretchy web-like strands, and those are actually the gluten strands. So if you think about it, there was no actual kneading in this dough, but gluten still developed. So why did that happen? Well, as I mentioned before, hydration is key in gluten development, so that's one aspect of it. And in addition to that, we also have more enzyme activity as the dough rests longer, because with more time, the enzymes in the flour and the yeast have more time to be active, and they can start to break down the starches and the proteins in the flour, which not only helps develop gluten, but it helps to give more flavor to the bread itself. So, and essentially, the enzymes are helping to break the proteins, the gluten proteins, into smaller pieces, which can help sort of align and form a gluten structure. In addition to that, we have the fermentation process, which is done by the yeast in the dough. And the fermentation process is releasing CO2, and those CO2 bubbles are actually sort of bumping into and moving the gluten strands. And that sort of gentle moving of the gluten strands actually helps them to sort of align and strengthen, if you can believe it or not. So you're not forming a super strong gluten network, but you are forming a gluten network with time in this scenario. And you can imagine it sort of as thinking of like maybe little armies of little teeny tiny yeast that are kneading the bread instead of you sort of aggressively kneading the bread. Instead, you have this sort of little army of yeast doing it for you in their own little special way. To visually show the difference between those two breads, on the left-hand side, we have that bread from 2 slides ago, which was that fast gluten development bread. So the total fermentation time was only about 2 hours, and this was a yeasted white bread. And so kneading was what helped speed up that process, and as a result, we got a very soft bread with really small air bubbles, very, very delicious, of course. And then on the right, we have the result of the bread from the previous slide, which had that long fermentation and slow gluten development with no kneading. And this is also a yeasted white bread. And an important thing to point out is that the ingredients are different between the two of these. The one on the left-hand side had like butter and sugar added, for example, whereas the one on the right-hand side was just the flour, water, salt, and yeast. And this longer fermentation one had a different texture, a little bit more chewy, and also the crust was a bit more crusty, which also has to do with the kind of baking style that you use for no-knead breads. And also, of course, there's bigger air bubbles respective to the fast gluten development. So I thought it was important to give a visual of what gluten strands can look like after the bread is actually baked. So here I have some sourdough white bread that I filmed while I was sort of tearing the bread apart. And as you can see, they have quite stretchy elasticity to it, which is those gluten strands that form during the kneading process and also develop further while you're baking the bread. And I just wanted to show these because they're quite, I don't know, beautiful and aesthetically pleasing. Bread is very, very, very sexy in my opinion. So to finish the discussion of the protein that's in flour and the importance of gluten, I wanted to point out that the amount of protein in gluten actually differs within wheat flour and within different kinds of flour made from different grains. So in the figure on the left-hand side, this is a box plot of different wheat flours, and as you can see, the amount of gluten and protein in this case, increases with different types of flour. Cake flour having the least amount of gluten. It's actually treated with a gas, I believe, in order to help break down that gluten structure. And this type of flour, as mentioned, is used in cakes to create that sort of light, soft, crumbly texture, as opposed to that tougher, chewy texture that you can get in really high gluten breads. So then as we go up the chart here, as we get up to bread flour, which is the most common flour used in bread making, that has higher gluten, which is good because it helps form that bread structure, which is very important for building the bread and creating flavor and structure in the bread. Then we also have a comparison of different types of flour, for example, wheat and rye and oat, and rice and millet and corn. The most common types of flour that you'll see in bread making, or at least that I see in bread making in my life, are wheat and rye, and the main difference between them being that it may not seem like it's much lower, but the amount of gluten content in rye flour is actually what I would consider substantially lower than wheat flour. If you make a bread that's 100% rye as opposed to 100% wheat, it's going to be denser, and it's going to sometimes be a little bit more gummy, and it's also not going to really hold its shape quite as well as wheat flour. It might be a little bit more crumbly because it just doesn't have as much gluten in order to build that sort of sturdy, stretchy, elastic texture that you want in a wheat dough that then transforms into a nice, sturdy, chewy bread. So enzymes are another very important part of the bread making process. They're found in flour and yeast, and they're also found pretty much everywhere. They're found in our bodies and our saliva. They help us to break down food. They're also found in the soil and pretty much everywhere that life exists, so you can find enzymes. And what they are is that they are proteins that help catalyze or speed up biological reactions by interacting with substrates. Substrates being the molecule that they're reacting with, and they can help sort of break them apart or bind them together. So essentially they're speeding up the rate of a reaction by binding with and enhancing the rate of the reaction. And here we have a diagram where we essentially have what looks like a little blue Pac-Man, which is the enzyme, and it's about to bind to substrate A. And the reason why it can bind to substrate A is because it fits into this enzyme's particular binding site, whereas substrate B does not. So it can help speed up the reaction associated with substrate A. So enzymes can actually theoretically work forever, as long as they have enough of a substrate to keep working with, and as long as temperature and pH conditions are ideal. But of course, if pH or temperature or substrate concentration gets to a point where it's not ideal, then this can interfere with enzymes' productivity. So enzymes are sensitive to pH and temperature, as well as the substrate concentration. So usually with pH and temperature, there's a range of pHs and temperature that the substrate is most active in, and that's usually sort of the peak or the hump of the graphs that we see here. And then there can be an actual optimal or like best pH or temperature at which the enzyme is the most active and most productive. And in terms of temperature, the reason why the enzyme tends to be less active at lower temperatures is usually because when there's less heat in the system, the molecules aren't bouncing off of each other and interacting as much. And so therefore it's less likely that the substrate and the enzyme will bind. Whereas with a little bit higher temperatures, when we get to like the peak of that hill there, then there's more heat in the system, so more energy, and the molecules are bouncing off each other more, so there's more of a chance that the enzyme and the substrate will interact and react and speed up that reaction. But then as you can see at the bottom of the hill, it starts to decrease quite drastically. And that's associated in terms of both pH and temperature with the fact that when the pH and the temperature get to be too extreme, it can actually start this process called denaturing, which essentially means that the structure of the enzyme itself starts to break down and it loses its ability to bind to the substrate. So it essentially stops being functional. And so then the reaction stops happening and the reaction rate decreases pretty dramatically. So overall, the pH and the temperature are not only important in these enzymes, but then if we apply that to bread, a lot of times when you're making bread and you're letting bread rise, it's usually in a warm environment because that's when the enzymes tend to be most reactive. But then if you stick it in the oven, those enzyme reactions stop at some point because they get too hot. And if you make it too cold, then the fermentation and the bread rising usually decreases quite substantially. So obviously pH and temperature are not only relevant in the enzymes, but it's also really relevant in the bread making process. So now we can start discussing the enzymes that are in flour and yeast. So the most important include amylase and maltase, which help break down the starch in the flour, as well as protease, which helps break down protein, including gluten, lipase that helps break down lipid, and then lipids in the flour, and then we also have the invertase and zymase, which are usually specific to the yeast, and I'll be discussing them later. Okay, so I drew this little diagram to show sort of the process of enzymes and their breakdown of the starches in flour. So we start at point A, which is the starch, and the starch is broken down by an enzyme called amylase, which can be found in both yeast and flour. And that breaks down the starch into maltose. And then maltase, which can also be found in yeast as well as flour, can help break down the maltose into glucose. And the glucose is important because that can be used in the process of fermentation, which is a process that is performed by yeast, which breaks down gluten into alcohol in the form of ethanol, as well as CO2. And the yeast use the enzyme zymase in that process. Another sort of tertiary or like secondary enzymatic process is involving invertase, which is an enzyme that can be found in some types of yeast. In particular, baker's yeast or store-bought yeast tends to have yeast that contain a lot of invertase. And they're able to break down sucrose, which is white table sugar, into its respective components, which are glucose and fructose. So to discuss amylase in just a bit more detail, it helps to break down sort of large, complicated starch molecules into maltose molecules, which are essentially two glucose molecules that are bound together. And then maltase, of course, helps to break down that maltose, which, as I mentioned, was two glucose molecules bound together, into two glucose molecules, which can then, of course, be used for fermentation. So as mentioned, bread really only requires flour, water, yeast, and salt. So why is sugar added to most breads? Well, that's because the enzymes in the flour and yeast take a lot of time to activate. So it takes time for them to go through that chain reaction of breaking down starch into maltose and then into glucose, which can be used for fermentation by the yeast. Whereas when you add sucrose, which is white table sugar, that is a very energetic and more favorable form of sugar, and it's comprised of 50% fructose and 50% glucose. And as mentioned earlier, that glucose is used in fermentation. And an enzyme that's often found in baker's yeast or commercial yeast is invertase. And what this can do is it can quickly break down that sucrose into glucose and fructose, and it speeds up the fermentation process and the sugar breakdown much faster relative to the amylase and maltase pathway mentioned earlier. So if we think about the two breads I mentioned earlier, we had the fast fermentation bread, which needed to be kneaded, and the fermentation time was about 3 hours. Then you also have the slow fermentation bread I mentioned, which didn't need to be kneaded, but it was rested for about almost 24 hours. So the fast fermentation one definitely had sugar in it. I added some white table sugar into the recipe to make sure that the yeast was activated in time. But the slow fermentation bread only included those 4 essential bread ingredients. And the reason that it was able to activate and ferment was because it was given so much time in order for those that longer chain of enzymatic reactions to occur, as opposed to the shorter invertase chain of reactions that occurred in the faster fermentation bread. So 2 other very important classes of enzymes, other than the enzymes that break down starch, include proteases and lipases. So what proteases are is that they're enzymes that are found in flour and yeast, and they help break down proteins, hence the protease aspect of the enzyme. So they help break down proteins down into amino acids, and then also lipases in the flour and yeast can help break down the lipids in the grain. So proteases are pretty important, and there's kind of a delicate balance at play, where you want some protease activity, but not too much protease activity. For example, proteases help influence the development of the gluten because they help to sort of gently sort of break down the protein so that the bread doesn't get too tough, and it helps the bread become more elastic as it rests and ferments. But if you let the bread prove for too long, then the enzymes can be too active for too long of a period of time, and they can start to break down the gluten to a degree where it can be actually harmful to the bread structure. And this is often something that occurs when a bread is overproved or rested for too long. The fermentation stops because the sugar has been used up, and then the enzymes that are breaking down the gluten are becoming sort of overactive, and they're really just sort of destroying the structure of the bread. And then because fermentation isn't occurring anymore, the bread just sort of deflates and becomes weak and can't really bake it anymore, or it's going to be a not so pleasant lump to eat in the end. Another important aspect of proteases is that when they do break down those proteins, which are the gluten, into amino acids, these amino acids actually really contribute to the Maillard reaction, which is a reaction that occurs between the sugars and the amino acids in the bread as it's baking, and they help with the browning and flavor development of the crust. So overall, proteases are very important to bread development, and you kind of want minimal activity in order to achieve good gluten development, but not so much that the bread structure starts to suffer. In addition to that, we also have lipases, which aren't really discussed as much, and I'm kind of starting to think that maybe they're not quite as important in the bread making process. Hard to tell, and they might have more of a factor if you're adding something like milk or butter or some sort of fat into your bread. So that's something that I could probably take some time to explore more. So just to reiterate a point made in the last slide, enzymes, whether they break down starch, proteins, or lipids, really help extract more flavor from flour. If you think about just like eating a spoonful of flour, it's pretty plain and very unpleasant, but you eat, a handful of bread and it's delicious, even if it just has those 4 basic ingredients. And that's because through the enzymatic activity that occurs during the fermentation process and the flour breakdown process, it really helps to extract more flavor from the bread. And part of that flavor extraction has to do with the Maillard reaction, as I mentioned in the last slide, where you have those sugars and amino acids that are byproducts of enzymatic breakdown that really help to contribute to the Maillard reaction, which also adds more flavor to the bread, especially in the crust. If you can see in the figure on the right-hand side, this is the same bread recipe, but at the top, I didn't bake the bread for as long as I did on the bottom. And as you can see, it's more brown, which actually means that the Maillard reaction was given more time to occur. And so you have a deeper color and also a deeper flavor in the crust. So I've talked about flour quite a bit, so now I can start discussing another very important part of the bread making process, which are yeast. And I've mentioned them previously in the process of fermentation and the fact that they share a lot of enzymes with flour, such as maltase and amylase. And so yeast are actually a single-celled microorganism, and they're actually a fungus, kind of like mushrooms. Commercial yeast, which is mainly what I'll be talking about today, are usually made including the species Saccharomyces cerevisiae. I might be pronouncing that incorrectly, but it flows smoothly out of my mouth, so that's good enough for me. And as mentioned before, they're very important because of the enzymes they contain, and we'll be talking about another important enzyme they contain in a little bit. And they're also critical for the fermentation process, which is very important for bread making. So what is fermentation? Okay, so fermentation is a really important process in bread making, as I've mentioned probably 500 times by now. It's a process that breaks down the glucose, as we mentioned earlier, which can arise from the breakdown of the starches in the flour or from a sugar source that you add to the flour. And the fermentation process essentially breaks down that glucose into ethanol, which is an alcohol, and carbon dioxide. And this process can only occur anaerobically. So that's usually what bread is covered, and there's usually sort of like micro pockets within the bread, which are these little fermentation bubbles that are sort of anaerobic habitats for the yeast to perform fermentation. And the reason why yeast are doing this fermentation is because they can extract energy from the glucose, which is very important for maintaining their populations and their biomass. And an important enzyme in yeast that performs the fermentation process is called zymase, and that's what helps break down that glucose into ethanol and CO2. And in the process of fermentation, you're not only making these end products, but you're also developing flavor in the bread itself, as well as creating gases, which help the bread rise. You can sort of think of the gluten and the gases in fermentation as a balloon of air that you're blowing up. You have the gluten, that's the actual like latex balloon itself, and then you have the CO2 bubbles and gas bubbles created from fermentation that sort of inflate that and stretch it out. And that's how you can consider bread to grow, whether it's rising or whether it's baking. And this little GIF on the left-hand side can sort of represent that. As it's rising, the gluten strands are sort of trapping that CO2 and expanding because they're stretchy and elastic, as I mentioned earlier. And that CO2 helps to sort of build the structure of the bread and to make it airy and light as opposed to super dense and chewy. And another note on fermentation, the reason why the yeast perform this anaerobically is because when there's a lack of oxygen, oxygen is usually a very good and viable energy source for various chemical reactions that are performed. And so when oxygen is exhausted and conditions are anaerobic, instead of oxygen, glucose is very, very favorable for energy usage. And so that's why fermentation can sort of kick in order to use glucose for energy and for these yeast to survive and to keep their populations growing. And then these pictures on the bottom are from a fast yeasted bread that I made. And we have the bread on the left-hand side at 0 hours right after it was kneaded, and then an hour later after it's been given time to properly rise, and it's over doubled in size from using baker's yeast or commercial yeast, which can ferment very quickly, therefore improving and increasing the size of the dough. So now to get into the differentiation between different types of baker's yeast or commercial yeast. As mentioned before, these are primarily the Saccharomyces cerevisiae fungus or yeast that are used in the mass production of these fast-growing yeasts. And they can come in various forms. As you'll see in the bottom right-hand side, you see the sort of weird, mushy, Play-Doh-looking block of stuff. And that is something called cake yeast or fresh yeast. This is not very common in the US or Canada, but I experienced it firsthand when I was living in Europe for a while. It's sort of this weird, it has the strangest texture, but essentially what it is fresh yeast, and you have to use quite a bit of it compared to a dried or active yeast. So I won't really be discussing that in great detail, but that's just one form of yeast, commercial yeast that's used in different countries other than the US and Canada. So then we get to the differentiation between active dry yeast and instant yeast. The main differentiation being in the size of the actual granules. So active dry yeast tends to be in larger granules relative to the instant yeast. And what that means is that you usually have to dissolve that active dry yeast in a little bit of water and sometimes sugar to sort of activate it before you actually make the bread. In order to dissolve the yeast colonies that are located within it and to sort of get them to be ready by the time they're added to the bread, as opposed to instant yeast, which is ground very finely, as you can see in the picture on the left-hand side. And because it's ground very finely, it can usually be added to the flour and mixed into the bread immediately without having to be activated first, because it's in such small granules that those yeast will just readily dissolve and start activating and start doing the fermentation process. So another thing I read about is actually called rapid rise instant yeast, which apparently is even faster than instant yeast. And not only are the granules of the yeast very, very small, but it's usually also mixed with enzymes and other additives that can make it sort of prepare the bread much faster than these other forms of instant yeast or quick yeast or commercial yeast. And so apparently with this rapid rise instant yeast, you can actually start to shape the bread and get it ready to bake like right after kneading, so you don't need to do 2 separate fermentations is what it appears to be. So, as mentioned earlier, the 3 main things that impact enzyme success include temperature, pH, and then the substrate concentration. And so, as you can see, the yeast, in this case commercial yeast, typically prefer a temperature range of about 80 to 110 degrees Fahrenheit or 26 to 43 degrees Celsius. But then they can die when temperatures exceed 140 degrees Fahrenheit, and that's usually because the proteins or enzymes within them start to denature and degrade. And so if we consider the different types of enzymes that can be in yeast or in flour as well, we have amylase, maltase, invertase, and then zyamase, which is that enzyme that's used in fermentation. All of their temperature ranges fall between that 80 to 110 degrees Fahrenheit range that yeast prefer, meaning that the enzymes and the yeast preferences in terms of temperature are very correlated. Another important factor about the temperature and yeast is that fermentation occurs best at that 90 to 95 degrees Fahrenheit range, which is the same range as zyamase's temperature preference. And every degree above that 95 degrees Fahrenheit threshold can actually start to impact fermentation negatively, because if temperatures get too high, then the yeast and the fermentation start to produce too much alcohol, and therefore it can make the bread taste a bit more alcoholic, and it can start to limit that yeast activity, which sounds sort of counterintuitive. If a yeast is producing lots of alcohol, you would think that would be okay, but no, it's kind of like if you're trapped in a room breathing out too much when you're breathing in. So you're consuming the oxygen, releasing lots of CO2. And at some point when oxygen is depleted, then the amount of CO2 that's built up in this small space where there's no new oxygen coming in, you could start to suffocate, which is way more negative of a topic that I meant to get to on this bread. But that's sort of a proxy for alcohol, too much alcohol building up after the fermentation process. It's kind of like too much CO2 building up if you're trapped in a room with limited oxygen. And then if we consider pH preferences, as you can see here, I posted the pH preferences, and these enzymes appear to prefer sort of more relatively acidic pH conditions. And if we think about why that is, it's usually because as sugars are being broken down during fermentation, the yeast during the fermentation process are releasing CO2, and they can also release organic acids, and then ethanol as well can also break down into acids as well, which is why the dough's pH tends to be more on the acidic range, and that's why these enzymes tend to prefer more acidic pHs. So, as mentioned before, zymase is very important in yeast fermentation. It's essentially responsible for the fermentation process. And as with any enzyme, as mentioned earlier, yeast and their enzymes generally prefer a temperature of around 80 to 110 degrees Fahrenheit. And so what happens if you decrease that temperature? What happens to the yeast? What's going on here? Because a lot of doughs, for example, pizza dough or sourdoughs, usually require a cold fermentation that lasts up to a few days even. And this can really, really greatly impact the bread's structure and flavor. And the reason why that is because when you're putting the dough into the fridge, almost all the enzymatic activity either stops or slows down very significantly. So the yeast are essentially less active and fermentation slows down. But then another little microbial population starts to come into play. And I call these, for example, sneaky bacteria, because when it's cold, bacteria are less inhibited by the cold temperatures as opposed to the yeast. And so the bacteria that are found in the flour and in the environment, and what they can do is they can develop some acids, which can sort of change the flavor and the texture of the crumb. So if we think about longer fermentation, which is really well represented by this pizza dough on the left-hand side, which was left in the fridge for about 3 days, and you can really see a difference in the color and the texture and of the bread crust that runs around this pizza. And so as a result of longer fermentation, this usually yields more flavor in the bread, or pizza in this case, more bubbles, because as the gluten relaxes during that cold fermentation, and therefore when CO2 arises, it can sort of hold more CO2 because it's more stretchy. It's like a big balloon as opposed to a small balloon. Another important thing that cold fermentation impacts is the texture. Sometimes it can make the bread a bit chewier or softer depending on what the recipe is. Okay, so the flour and the yeast sections were quite lengthy, but the water and salt sections, respective to those other sections, will be quite short. Which doesn't necessarily mean that they're less important. Water is very, very important in the bread making process because it influences a lot of important things. For example, I beat this to death earlier, but hydration and water are very important in gluten development and formation, as well as its extensibility and relaxation throughout the fermentation process. In addition to that, water can help sort of move around the enzymes, because when you're mixing the dough together, water is a fluid, and so it can help sort of distribute the enzymes throughout the dough. Another important thing that it does is that it hydrates the starch in the dough, which is very, very important when the bread is baking, and I'll be discussing that in the next video about starch gelatinization and what's happening there with starch hydration. Oh, and as mentioned earlier, it helps the enzymes disperse. It also helps the salt disperse in the flour. For example, if you dissolve the salt in the water first before mixing it into the dough, then the salt distribution is going to be much more even throughout your dough. Another important thing that water does is that it vaporizes during the baking process, which releases more gas bubbles. So not only are you getting gas bubbles in the bread because of the CO2 from fermentation, but you're also getting water vapor and gas bubbles that are released during the bread baking as well. So in the figures on the left-hand side, we have different percentages of hydration. In the top top left picture, we have 50% hydration, 68% hydration, and then 80% hydration. Most quick yeasted or fast breads are at about the 60 to 68% hydration. So they tend to have a very sort of consistent, not very bubbly crumb structure, not very open, not lots of big bubbles, as opposed to 80% hydration. And this example here in the 80% hydration in this top left image is very flat, showing that when you hydrate it more, it tends to relax more and you can get sort of more of a pancake-like bread. But that's not always the case. If you develop the gluten properly, it can hold the structure much, much better. So you can definitely get 80% hydration doughs that are very well structured and have really big air bubbles. As I mentioned in the last slide about long cold fermentations, if you're adding more water, the gluten is even more hydrated and it can help it relax, and therefore the air bubbles can be bigger and more pronounced in a higher hydration bread. Okay, so now to consider salt, which is the final very important aspect of the bread making process. Even though it's often thought of as just a flavor enhancer, it's actually much more complex and important than that. Just to explain the gluten structure a little bit, oftentimes gluten aligns in strands, and those strands are actually negatively charged, so they tend to repel each other, kind of the corresponding ends of a magnet. When you try to put the north and north poles together, the south and south poles together, they will repel each other. That's kind of what's happening here with the negatively charged gluten strands. And so when you have salt, which is sodium chloride, and that becomes dissolved in water within the bread, the sodium ions, which is that Na+, can bind to those negatively charged sites, and it can actually help gluten strands stick together, because when you don't have those negatively charged sites and you have some positively charged sites, it can help with adhesion, and that process helps to strengthen the gluten. So salt is actually pretty important in the bread making process. And another important thing is that the percentage of salt that you add really makes a difference in that gluten development. For example, we have the picture on the left-hand side, which has 0% salt, 2% salt, and 5% salt. 0% salt is considered too little salt. You don't want to not add salt to your bread, because it can make the gluten more weak and more cake-like, so you can get a more crumbly, sort of weak structured bread. 2% salt is considered that sort of Goldilocks stage where the gluten development is good, the flavor is good, and the influence of salt on the bread is at its sort of maximum potential. But then you also have 5% salt, which is just too much salt. Not only will the bread taste too salty in a lot of cases, but the gluten structure will get so strong from adding so much salt that it'll actually become too tight and dense. So 5% is too much, 2% is good, and 0% is too little and not recommended. Another important thing that salt does is that it can inhibit or slow down enzymes. And this can actually be a good thing, because if you're slowing down enzymes, then it can actually allow more starches and sugars to build up in the bread, because those enzymes aren't able to act on them in time, which can actually help contribute to the flavor and browning of the bread as it bakes. And similar to that, salt can also inhibit the yeast. Not only does it inhibit the enzymes in the yeast, but salt can also cause osmotic stress for the yeast and potentially kill it if there's too much salt. So in doing so, because it slows down the enzymes, it can also slow down fermentation, especially if it's killing yeast. Then it's certainly going to slow down the fermentation, and you could potentially ruin your bread if you add too much salt. Which is why, in a lot of cases, 2% salt is ideal, because it enhances flavor in a good way, it enhances gluten structure in a good way, without having too many adverse effects. So as mentioned, it really plays a big role in the flavor development, and in addition to that, salt also helps preserve the bread after baking. For example, it can drive away certain microbes and molds that might be attracted to the bread, causing it to turn bad quicker and become moldy faster. So it helps preserve the bread, which is obviously a very good thing. So just to wrap up this long-winded presentation about the science of bread, I wanted to point out the main points and the conclusions of this presentation. So as mentioned, flour, water, yeast, and salt are essential for creating good bread. You can add bells and whistles like butter or oil or milk or sugar, things like that, to enhance the flavor or the structure or the color or anything you want from the bread, essentially. But those are the essential 4 ingredients for making good bread, because they play a big role in the structure, the flavor, and the texture of the bread, and the success of its rise as well. And then we can consider some major points that really influence the bread's success in the bread making process, and that includes proper gluten development, which requires water. Another important aspect of proper gluten development is the aspect of time versus kneading. For example, that quick rise bread versus the slow rise bread, as well as leaving it to ferment cold overnight. in addition to that. And then also the critical importance of enzyme activity, whether it's the ones that are in flour and yeast that are involved in the breakdown of starches or proteins, as well as the enzymes in yeast that help with the fermentation process. We also have the temperature and pH, which are really important factors that influence the activity of those very important enzymes. And then also the impact of salt and water that are important for gluten development and flavor of the bread and structure of the bread as well. So in conclusion, I hope you enjoyed this presentation. I know it's not quite as in-depth as it possibly could be, and for some people it's probably more in-depth than you ever really wanted to know, but I really just wanted to get kind of a basis for bread science down. And as you can remember, this presentation was mainly about commercial yeasted bread. And a lot of these factors will change when you start discussing a long fermentation or sourdough bread, which I might be making a video about in the future. So I hope you enjoyed this presentation. I hope you did learn something or possibly many things, and have a great day, and perhaps you will see me again soon.